So it was just like some kind of trouble with the Zoom. So I think now everything is fine. So first of all, I would like to introduce myself. My name is Mohamed Elhadidy. I'm a professor of biomedical sciences.
I don't think we've started, Mohamed. Have you guys let everyone in to the room yet?
Okay.
Yep, everybody is in so far.
Oh, okay, excellent. Sorry, I'll introduce you, Mohamed. It's fine. Is everybody able to hear and see us? Just point out in the chat that it's all okay, and we'll begin the session now. Sorry for the delay. Hello and welcome to the Public Health Alliance for Genomic Epidemiology webinar.
Nabil, can you just give me 1 minute, please?
I'm a senior bioinformatician. Yeah, sure.
Okay, just a
minute. So yes, I'm a senior bioinformatician at the Quantum Institute here in the UK, and I'll be a chair and moderator for today. We had a bit of technical difficulties in the last few minutes, and hopefully everyone can hear and see us. Our speaker, Mohamed, has just joined us. He's just snipped out for a minute, and while he's away, I'll introduce today's session. So today's session, it is 1 hour. And we'll have the speaker present to us for about 45 minutes, and then we'll have a short segment at the end for questions. If you have any questions, please post them in the chat as we go along, and I'll relay them to the speaker during the Q&A at the end. Our topic for today is One Health Microbial Genomics Approach for Epidemiological Tracking of Bacterial Pathogens along the farm to fork continuum. And today our speaker is Dr. Mohamed Elhadidy. Dr. Elhadidy is currently a professor of biomedical sciences at the Zewail City of Science and Technology in Egypt. Dr. Elhadidy holds a DVM degree from Mansoura University in Egypt. He obtained his master's in 2006 in microbiology from Mansoura University and obtained his PhD in microbiology from Virginia Tech University in the United States. Dr. Elhadidy's research interests include pathogenesis, ecology, evolution, and transmission of bacteria that cause foodborne and zoonotic diseases. And he uses a number of different population genetics and comparative genomics approaches. His group has also applied sequencing data to genome-wide association studies to explore the association of different accessory genes and core genome SNPs within biofilm formation, this being a key facet of antimicrobial resistance. So we started off to a rather shaky start, but it's good to see you, Mohamed, and we're very happy to start now and listen to your presentation.
Thanks, Nabil, so much for giving me the floor. And my apologies again for being a bit late. I was having some technical issues with the Zoom, so now everything is fixed. So as Karim mentioned, I'm a veterinarian by nature. So I spent I studied veterinary medicine at Mansoura University, and I studied also microbiology at Virginia Tech University. And my main passion is usually about microbial genomics in order to determine the bacterial evolution. So I'm a bacteriologist, and I'm having some insights into how to use the microbial genomics tools in order to study the evolution of different bacterial pathogens. Okay, actually my presentation today is dealing about applying a One Health genomic approach, being a veterinarian, and what I mean by One Health is how to integrate different healthcare disciplines to study the evolution of different bacteria and for epidemiological tracking of different bacterial pathogens from the source or from the barn to form continuum. As I mentioned, my main interest is about studying the bacterial evolution. And if I want to study the bacterial evolution, my main example or the clear example is the evolution of antimicrobial resistance. So in 1928, penicillin was actually discovered by Alexander Fleming, and it went into the developing phase. And here the question is what happened in a decade from 1928 to almost a decade to 2022 now, that this bacteria changed from being very susceptible to most of the antimicrobials into being a pen resistant. And most of the people are talking today about in maybe in like 20 minutes or so, we are into the pre-antibiotic era. So what I mentioned here is like, this is a really clear example of studying the bacterial evolution, is studying the bacterial antimicrobial resistance mechanism. So actually I'm interested into studying these mechanisms in genomic approach. That's why we name it like, while searching for the hero, we created the monster. And I have to highlight here that, we didn't really create the monster. It's just like some kind of making some kind of dramatic, caption for it. Actually, like the bacteria are evoluting, but maybe the selective pressure of the intensive use of antimicrobials actually made the bacteria more resistant, but actually bacteria is evoluting actually naturally. So here, if we actually we want also to see the timeline of this disaster, as I mentioned, in 1928 with the discovery of the penicillin, and let's say in maybe 25 years later, which 1950 to 1960 was a golden age of discovery of most antibiotic, as people were saying, okay, we finally find a cure for all microbial infections. And by then, with the intensive use, and now, as I mentioned, bacteria acquired a lot of resistance, and our role here is to make a surveillance study in order to determine the molecular mechanisms of different resistance. And we know that this resistance are mainly, if I'm talking about epidemiologic, about the intensive use of antibiotics, especially in the clinics, okay? And what I mean here also that, these types of crisis is not actually confined into a single area, because of our traveling. So what we mentioned here, that a risk anywhere is a risk everywhere. And because we travel, and if we recall, one of the main resistant mechanisms for antibiotic, which is New Delhi metallo-beta-lactamase, and it was named so because it was actually first reported in a British patient who actually traveled to India, and then he brought this antimicrobial resistant gene. It's carbapenem resistant gene. So what we meant here that, with the international travel, this crisis might be actually augmented and increased. And people are actually expecting that by 2050, the crisis of antimicrobial resistance might be more risky than, because we are inventing more grants and more money on cancer cure, but simply people can die from simply diarrhea. So we have to, to tackle this as well. Also, if we, in Egypt per se, or one of the developing countries where was still prescribed, we don't have a prescription. You can get antibiotics on the counter, and that actually is a scenario in different developing countries as well. So here the question is, if we stop getting antibiotics from the counter, and prescription is really crucial in this, should antibiotic resistance really decrease? And from the One Health approach, maybe it's not. Why? Because actually also we are using antibiotics at the farm, in the animals. So also in Egypt, we still use antibiotics as a growth promoters. And when we use antibiotics, we eat this food, so we can acquire this resistance not by by using misuse of antibiotics in the clinics, but also from eating of food that is contaminated with antibiotic resistant bacteria. And that's why the main concept of the One Health. So One Health is integration of different healthcare disciplines, environmental engineers, veterinarians, public health sectors, epidemiologists, and physicians, but it's also integrating the integration of different, as I mentioned, health risk anywhere might be a health risk everywhere. So that's why we are collaborating, to discuss the potential solutions, and that's what we named act local but think global as well. Okay, so this is my main approach. So I'm going to present in here this type of One Health approach that I'm implementing since my return in Egypt 5 years ago in my own institution, to study the surveillance and the epidemiological surveillance of different foodborne pathogens from the farm to fork using the One Health approach. And one of the model organisms that I'm working in is Campylobacter, and it's not really a new foodborne bug, but it's like, it was discovered in 1975, and before then, most of the common foodborne pathogens that are actually zoonotic are Salmonella, Shigella, Escherichia coli, etc. So no one was knowing about Campylobacter before 1975. Yet by now, I would say that the Campylobacter is the important and one of the most predominant foodborne pathogens, and the incidence or the infection rates of Campylobacter is actually more than Salmonella and Shigella combined. Okay, so if we can how can acquire Campylobacter, we know that Campylobacter is actually like a foodborne pathogen, as I meant. It's mainly through eating uncooked poultry. So if you eat like chicken that's uncooked, you might be at risk of developing Campylobacter. So this is actually the transmission cycle. As I mentioned, it was known before that mostly you can acquire Campylobacter by eating uncooked chicken, yet due to bacterial evolution, due to what we named spillover host, we have different reservoirs for infection. And what I meant of a reservoir, that's something that the bacteria can depend to enrich before being transmitted to the human. So we have different animal reservoirs, not only the poultry, cattle, swine, sheep, pets, all this, and wildlife as well might be a potential source, not only the chicken. So you acquire this by eating through the farm to fork, by uncooked unpasteurized milk, or uncooked chicken or so. And also we have some kind of environmental rules of transmission of Campylobacter, maybe the drinking water, maybe the irrigation water, etc. So we have now some kind of a complex epidemiology of Campylobacter, and the bacterium is rapidly evolving. So now the common sense that this bacterium is an open genome that I will mention later, and it's evolving everywhere. So there is no common reservoir for the infection, as before we thought that the chicken is the main reservoir. So applying the One Health approach, we can use the microbial genomics in here to understand, or to better understand this transmission cycle. How can microbial genomics can aid? And before applying in this is really before having promoting for the microbial genomics, or how we can use microbial genomics, we can have ever imagine before the era of the microbial genomics, what the typical form of understanding the transmission cycle. Okay, so we know that evolution is because of the basic biology is evolution is bugs can evolve because of two things. First of all, clones, one clone that might have some kind of dissemination success. So evolution wise, it's better fit for the environment. And that the basic fit, the basic theory of evolution by Charles Darwin, that the fittest might be better evolving, or or the less fittest might be at risk of to perish. Okay, so the basic biology is the clones and the horizontal gene transfer. And what I meant here by horizontal gene transfer, that bacterium, because it can easily acquire foreign DNA from other bacteria, etc., by a horizontal gene transfer, either by conjugation, transformation, and transduction. So in order to determine the clones, usually the gold standard, or the standard that we commonly use to characterize the clones in Campylobacter, the gold one, is called the multi-locus sequence typing. And in this multi-locus sequence typing, as you see in here, this is a bacterial genome, which is the genome of the Campylobacter is tiny one. It's 1.6 million base pairs. So you select 7 housekeeping genes. Housekeeping, it means it's really crucial for the metabolism of the bacteria, and it's conserved among all the genomes. So you select 7 of these genes, and you do a simple Sanger sequencing. And by doing simple Sanger sequencing, you can have different alleles, and the alleles can assign this bacterium into some sequence types and clonal complexes. And by implementing this technique, you are usually targeting these 7 genes and do a simple Sanger sequencing. And what I meant here, if you sum up all these genes, you are getting 3,300 base pairs, which is almost 0.2% of the genome. Okay, so this is the gold standard before the era of the microbial genomics. Okay, so this is for the clone. So you determine the clonal complex, and you you draw a phylogenetic tree or so using the common genetics tool, which is Microbial, multi-locus sequence typing. And for characterization, either for like you collect like all the antimicrobial resistant genes from the like literature reviews, and like you have your bug and do like a phenotypic testing, and then once you do the phenotypic testing, you do a DNA extraction and do a PCR, like a PCR for all resistant determinants, and if there are some alleles or like anti-grown cassette or so, you do a more Sanger sequencing. So it's more into a PCR and a Sanger sequencing, and this is like the main mechanism, we currently, like before, like 2 decades ago, everyone used to do in the lab. Okay. Yet that's like, and what I meant, like, I have done this myself in understanding the Campylobacter. I used to be a research associate in Scientific Institute of Public Health in Belgium, and we use this. Okay, so we have different, like, like publications in understanding the Campylobacter using the multi-locus sequence typing, and this is like a minimum spanning tree. It's a One Health approach as well. So the red ones are like the way, as you see, like you have two kind of circles. The red one is human isolates, and the green one is poultry ones. Okay, so if you have a circle like this and you have a clone that's in poultry and human, this means that this clone might be a high risk of infection to humans. So the more you can have from, like, like the animal reservoir, and you compare the minimum spanning tree, and you have some kind of a nice clustering like this by the old gold standard, which is the multi-locus sequence typing. Also, like, like you can have like some kind of insight based on literature review. You do, as I mentioned, the characterization, and you do like the Sanger sequencing, and from here you can have some kind of handful of resistant determinants here for like the erythromycin resistance, which is like also one of the gold antibiotic still used for treating Campylobacter. But you can have some kind of mutations in the erythromycin resistant determinants as well, and also in the efflux pump. So I mean, like, this well-known method is actually like it's fine, and like, like we have used this like maybe 10 years ago in characterizing Campylobacter resistant determinants, and also in understanding the epidemiological history of this bug. As I mentioned, like, 5 years ago, I started to build my own lab in Zewail City for Science and Technology in Egypt. And, like, I think like I went there with a different perspective, moving from the gene level, like the old school, to the genome level, and actually, like, I was lucky enough that we have a next generation sequencing facility, so we can do this ourselves. We have good collaborators as well that I will mention later. And what I actually want to emphasize here, that moving from the gene level to the genome level, like it's more upscaling, in understanding the basic epidemiology and like the transmission networks of this bug. So it can determine antimicrobial resistant genes as well, but instead of having like, compared with previous literature, you can actually compare with databases, like, like web-based databases such as like the CART, the Risk Finder, the Abricate, the Pathogen Finder, and all these of database are database for all antimicrobial resistant determinant. So you actually like do, and I will show you the workflow that we do. You can determine the microbial ecology, or like the transmission cycle from the animal to the human, because I'm gonna also mention it like, like maybe in a minute or so. We also like the good insight in microbial genomics, or like, moving into the genomic, that you can have some kind of genetic signature. Like every reservoir can have some kind of like allelic genetic signature that will use later in a source attribution analysis. So if we can have this kind of genetic signature that is unique among different reservoirs, that can be used to track what are the potential source of infection. If you have this genetic signature, say, hey, okay, might be this is like, the cattle is a source for infection, or the poultry, and we're going to show you this. As I mentioned, in multi-locus sequence typing, you do like a sequence for like 3,300 base pair, which is like 0.2% of the genome. Yeah, in microbial genomics, you can have like the whole genome sequencing, or like it's a draft genome. So the more coverage, the more like it can cover the whole genome. And what I meant by the whole genome, we name it in microbial genomic a pangenome. And a pangenome is the whole genome, and it's usually divided into a core genome. And what I meant here by core genome, that it's conserved among 99 to 100%, okay, of all the genome. Can you imagine, like, in the multi-locus sequence typing, the core genome are just like 7, like, the core genes. Here, you can make upscaling, and in Campylobacter, we can actually use a core genome into 500 or maybe 700 of core genes, okay? We, as I meant, like pangenome is like core genome, which is like 99 to 100%. Soft core genome, and what I mean by soft core genome are like 95 to 99%, and the rest we name it accessory genome. And the accessory genome are mainly genes that are acquired mainly by horizontal gene transfer. So what I meant by here, you can have an upscaling, of the core genes, stuff like just 7 genes. You can go into hundreds, maybe thousands, in some bugs of like characterization of this core gene. So it's more or less like an upscaling, and also it can have a better resolution and higher discrimination for the bacterial cluster. So these are the good insights in moving from the microbial genetics to the microbial genomics. So as I mentioned, like that was our hypothesis, like, like what we aim for, and luckily we have succeeded in having some kind of like very good insights. Okay, so this is a workflow that we used to have. Like it can be applied actually. Like this workflow can be actually like implemented like across different bugs. So we do a sequencing. We assemble, like, because we are short read sequencing. So you usually assemble this, like, as a puzzle. We usually use spades. Yet our experience that spades are not like, the top-notch approach. We can use a unicycler as well. And we do like, annotation by Proca or like Scori or Rory and stuff. And then we do a typing. And here is like, the click. Instead of doing a typing by multi-locus, we can still use like the in silico multi-locus sequence typing, but it's better here to make a typing based on the core genome multi-locus sequence typing. Instead of the usually 7 housekeeping genes, you can have maybe hundreds or thousands. So here is a higher resolution and higher discrimination power. You usually, like, what I mean, I usually say to my students, like, I won't discuss anything related to evolution. Instead, show me the tree first. So we do like the phylogenetic tree and minimum spanning tree in order to see the clustering. And then we can determine like the antimicrobial resistance based as compared to the public data set. So in one click, you can have just like, instead of doing like typing and characterization and PCRs and PCR, just in one click, you have the contigs or the raw reads. It depends on the pipeline you are using. You can have like a very nice Excel file of all the predicted in silico predicted antimicrobial resistant genes. As I mentioned, like, because whole genome sequencing is having a really good insight in having a genetic signatures. So also you can have a source attribution matrix that I'm going to show you later. Using these pipelines, you can have like divide your analysis into like a phylogenetic tree. If you are checking epidemiology, if you are typing, you can use like the pangenome, as I mentioned, the core genome, soft core genome, or the pangenome analysis. You can actually determine like the transmission cycles, as I mentioned later. Also, you can determine the recombination hotspots. So in recombination, we know that we have 2 different bugs according to the evolution. Like the bugs with an open genome that is actually horizontal gene transfer are actually making like, adding more genes, more accessory genes, and a closed genome. Campylobacter, like by the way, is an open genome. So it actually mainly acquire resistant determinants by horizontal gene transfer. Yet, like, we have other bugs such as the Mycobacterium. It's like a closed genome. So here, like, with some pipelines, you can determine, like, what are the hotspots for recombination. Actually, you can determine the recombination to the mutation rates. This is like the genome solution studies. I'm like, I'm not sure if you came across this, like, this types of study, but it's actually like, it started with like the eukaryotic genome. And what I meant, like, applying the GWAS, which is the abbreviation of the genome-wide association studies, actually, like, in my insight, it's like a revolution in like studying the microbiology. So usually in microbiology, we study the phenotype and we play with the gene. You mutate the gene and then compare the mutated versus the wild type, and then you complement what we name it, like in microbiology, the molecular version of Koch's postulae. With the GWAS, you can sort out different bugs based on genomic content, and then after sorting out according to the genomic content, you can verify what are these genomic content are actually in silico predicted in doing, and then you verify phenotypically. So in GWAS, you start with the genotype and then you go with the phenotype. The ordinary microbiology, like, we do the phenotype and then do the genotype. So again, like, these are really good insights when studying the microbiology, microbial genomics, especially at the One Health perspective. You can have very good insights, and it can give you a better epidemiological information about the zoonotic pathogens. So these are like the output of our hypothesis that I'm proudly with my team want to to announce. Like, we have like very good results, and I think this is one of like the publication I'm like personally I love. For some reason, like my name should be here, but like I think I just cropped the whole list of authors, which is like a second line as well. So it named like stress-resistant associated multi-host transmission and enhanced biofilm at 42 among hyper-irritant generalists Campylobacter jejuni. Like the title, it seems a bit complicated, but in theory, we know that the Campylobacter is really like sensitive to oxygen. Okay, so we called it's a microaerophilic, so it needs very tiny amount of oxygen in order to survive. This is the biology of the bug. Okay, yet we actually try to stress this bacteria. Okay, so this is a normal transmission cycle. Okay, so what I mean here that the bacterial agent, in order to get into a susceptible host, it actually face some kind of like environmental stress, or mean like if you have like a poultry or unpasteurized milk, you can have you actually can buy this milk from the refrigerator. Okay, or it can actually can be stressed by different like refrigeration or like temperature, oxygen, whatever stressors is. So we actually tried to figure out what are like the stressors that are actually, what are the stressors actually encountered for Campylobacter. And as I mentioned, that the Campylobacter is really like very sensitive to environmental stressors. So we thought like maybe this bacterium had made some kind of evolution and it's more resistant to different stress conditions. So we stressed, or we subjected this bacterium to different stress conditions, like the oxygen, which is aero tolerance, refrigeration and freezing, heat, chemical contamination, and osmotic stress, sodium chloride. So we subjected our bacterium to all these stress conditions, and we actually thought that maybe our bacteria have evolved several stress condition mechanisms, or maybe what we name it cross resistance. So what I mean by cross resistant, that resistant to one of these stressors can trigger the bacterium into being more stressful or more resistant to other stress conditions as well. And we also characterize the biofilm, and the biofilm is like an extracellular matrix like produced by the bacteria that makes it more resistant to antibiotics and also to stress conditions. So our hypothesis here that we studied the bacteria and we sort them out according to oxygen tolerance. Our hypothesis that the bacteria who are actually, we named them host generalists, and host generalists are, I mean here by the clones that actually can infect different reservoirs. It's not only two expressions we use in microbial ecology. Host generalists are like broad reservoirs, like a lot of reservoirs. Yet host specialists are actually reservoirs that only can infect one reservoir. And this is by the clonal dissemination success that I just mentioned earlier. Okay, so these are our results. We sorted out our bacterium based on three categories. HA8 means it's hyper-elotolerant. Hyper-elotolerance means that subjecting this bacteria to oxygen stress for more than 24 hours, it still can survive. Aerotolerant means that subjecting this bacteria to oxygen, it can survive up to 12 hours. And aerosensitive is just like normal Campylobacter that I mentioned, that it oxygen is really toxic for this bacteria. So, these are the kinetics or the time points. As you see here, we found different hyper-elotolerant bacteria and aerotolerant bacteria. And the nice thing here also that these hyper-elotolerant bacteria are also more what we named it earlier, like the cross protection, which is that these hyper-elotolerant strains are also resistant to different stressors. So we saw here it's more resistant, more capable of doing biofilm, and also that these hyper-elotolerant are more resistant to refrigeration temperature and to freeze temperature. And you see and heat stress. So being a hyper EO tolerance, it can give you a survival advantage of doing more biofilm and also in doing a cross protection or to have a more resistant phenotypes to other stress conditions. So here we thought that this is a high risk stuff, or Campylobacter, this hyper EO tolerant. And to study this, like, this is a phenotype, and to study this at the, again, at the, in studying evolution in bacteria, you sort out clones and you study the horizontal gene transfer. To sort out clones, we name bacteria into a generalist, I'd say like the clone that can actually infect different hosts or reservoirs, and the specialist which are not. So we found that host generalists are also more capable of doing, of being more resistant to different environmental stressors. So if we link this, like these generalists are actually this bacterium that can actually survive different stressors and are more capable in infecting different reservoirs. So if we link this slide with the slide that I mentioned before, that this host generalist are the hyper EO tolerant strain that are cross-protecting. So being this a successful clone that is making the bacteria more capable of making a cross protection and in making it more resistant, and this can give this bacterium a survival advantage over different other clones in infecting different reservoirs in Campylobacter, and for so in dissemination of the infection. So this was a paper that we published last year in 2021, and it's like, as I mentioned, it's like kind of a microbial ecology using some kind of insights of our sequencing of 112 Campylobacter isolates from different sources. We had like Campylobacter from cattle and also from chicken, and as well as from human infection. So by comparing, so we have some kind of microbial ecology study about the route of transmission of these bugs from the agent into like the cycle until it infects the human. Also, this is another paper we published last year from this Campylobacter microbial genomics project. And as I mentioned earlier, one of the insights that we're really interested in is having some kind of genetic signatures per reservoir. So if we have some kind of like genomes from different reservoirs that are potential sources of infection for the human, can we have some kind of genetic signature? Like this is like some kind of an allele that for cattle, this is like allele for pets, and this is an allele, etc. Okay, so this is like, a source attribution study using a model which like most of the people are using, which is a structure software model. And in here we have like different characterizations, as you can mention. We have different characterization methods. First of all, this we compare this characterization method, the core genome multi-locus sequence typing, and like I'm sure now, like you understand what the core genome, what the core genome is. The multi-locus sequence typing, the usual like seven genes. It's in here, but the core genome are like hundreds of genes. The host segregation markers that we actually found, or the genomic signatures per reservoir, comparative genomic fingerprinting technique, the normal multi-locus, the 7 multi-locus sequence types, and ribosomal multi-locus sequence types. So we compared all these typing mechanisms for their discrimination, and we found that the core genome and the host segregation markers are the potential, the highly discriminatory typing mechanisms, okay, that can actually, like, this is again based on genomic signatures, and this is based on core genome multi-locus sequence typing. These are the gold standards for typing of Campylobacter from different reservoirs, as compared to the, like 2 decades ago, the multi-locus sequence typing, which is actually giving a very low, like a relatively lower resolution compared to the other typing mechanisms. So we now need to shift from the normal one into this core genome or host segregation marker that are actually can be only revealed by doing a whole genome sequencing. So this is a discrimination power, and this is a source attribution analysis that we made by structure. And in the good thing in the structure that it actually estimates this allelic divergence among different reservoirs, and it's pretty simple. Like, we actually use these typing methods: the core genome, the host segregation, the MALST, and the CGF40 that we use to compare the discriminatory power. And this is like blue; it means like it potentially chicken is an infection source, and beige is like the cattle. Before doing this structure, we made some kind of verification analysis by structure as well, by doing self-attribution analysis. So we have isolate that we know it's from chicken and isolate that we know it's from cattle, and we do some kind of self-attribution analysis in order to verify the accuracy of this model that we named. And you can see here that you can use all these typing methods using the data from the whole genome sequencing results in order to predict what are the potential sources of infection. So if you have like a human infected with Campylobacter, by doing this type of typing mechanism, you can predict, or you can say to the patient, hey, like, you know, like maybe you ate like uncooked or unpasteurized milk, and this is a potential source of infection based on this typing schema. So I think this is really crucial in epidemiological surveillance and sourcing, tracing back of infection for human host. As I mentioned, there are like many trends also in genomic sequencing, and as I started my presentation by antimicrobial resistance, which I'm actually like interested in different bugs. This is also a nice insight and a paper that we published last year as well about implementing the high throughput sequencing and determining a more or a deeper resolution of genetic determinants in antimicrobial resistance in Campylobacter from farm to fork. We know that the Campylobacter, by the way, is self-limiting, and by adding a selective pressure to this bacteria, it's more resistant to antibiotics. It's not still a pen resistant, but it's still highly resistant to different antibiotics. So these figures are actually like one for a Campylobacter. These are the two main players in Campylobacter, Campylobacter jejuni and Campylobacter coli. So we made, again, this is a phylogeny based on the core genome multi-locus sequence typing, upscaling of like the normal multi-locus sequence typing, and all these rings represent the sources, the antimicrobial resistant patterns, etc. And as you see, like, you know, we can actually, like for Campylobacter jejuni, we can compare or we can build like three clades, A and B and C. And what we notice here that different clades have a homogeneous resistant pattern, which means that each clone have a kind of dissemination success in acquiring antimicrobial resistant genes. So as I mentioned, like doing whole genome sequencing and like microbial genomic analysis, it can give you a better insight about the evolution of antimicrobial resistance by having an upscaling of the typing mechanism, and by comparing this with the normal phenotypic data, you can have a better insights about the evolutionary mechanisms of resistance. So this is like our paper that was published also this year in 2022. It was in collaboration with our collaborators in University of Bath in UK. And this is something that we name it phylogeography. And phylogeography is like, okay, like if there is any genomic differences between our Campylobacter, which we isolated from Egypt, as compared to Campylobacter, which are actually isolated in UK or maybe in the US or so. And this is like, as we mentioned, we are usually interested. And if you recall, like my first like slides in my presentation, that One Health is not only about like collaboration between different disciplines, but also collaboration between different disciplines worldwide, because a health risk is anywhere, can be a risk everywhere. So here we try to use microbial genomics to order to have a better understanding or a deeper understanding of the evolution of Campylobacter in Egypt as compared to UK or Oxford datasets. And Oxford datasets we choose because it's a highly rich dataset that they have like a lot of genome sequence. So our hypothesis here that maybe the selective pressure in Egypt by using antibiotics in like uncontrolled use of antibiotics might have a selective pressure in acquiring and making horizontal gene transfer, or acquiring more accessory genes by horizontal gene transfer. So first of all, we made our tree, and as I mentioned, like our tree is the first, like, you know, any analysis about the evolution, we usually make a tree, and this is like a minimum spanning tree of like the red dots are the Egyptian isolates, and the blue ones are the UK datasets, and the gray ones are the global ones, and these are the different infection sources. You can see this is in jejuni and this is in coli. You can see that like Egyptian isolates are very clustered based on core genome. So based on core genome, our Egyptian isolates are really clustered well with the global datasets, the Oxford and the global ones. So it means that based on core genome, there is no big difference between the Egyptian one and the global one. Yet, by having some kind of like, this is like an analysis by comparing the accessory gene. This is a heat map for the accessory gene content of the Egyptian isolates as compared to the global ones. And we made, this is a core genome. Again, this is a comparison of average nucleotide identity. And this is like the pairwise accessory genome acquisition or comparison between the Egyptian isolates as compared to the global ones. And by doing this type of analysis, we have also, like, we compared between the accessory genome acquisition between country, like in Egypt, and within countries, like this is within the country and this is between countries. And you can see that the accessory genome within the Egyptian datasets is much more as compared to the UK datasets and the global datasets. Yet there is no big difference between the accessory genome acquisition between countries. And that actually means or supports our hypothesis that the accessory genome acquisition, by adding a selective pressure of using antibiotics, it might happen that there is more acquisition of accessory genome among Egyptian isolates as compared to the global ones, especially if we compared the antimicrobial resistant rates of the Egyptian datasets. It was significantly higher than the antibiotic resistant rate among the global datasets as well. This is also one of, like, this is a totally, so if you can, like, most of this like the publication that I just mentioned over the last, like, maybe 6 or 7 slides is actually like kind of like combining phenotypic results with the genomic data and trying to have some kind of an association between the phenotype and the in silico pre-reduction. This is a typical in silico pre-reduction of the toxin antitoxin system. So the toxin antitoxin is one of the efficient bacterial resistant determinants to stressors and also to antimicrobial resistance. So I just like emphasize different mechanisms of antimicrobial resistance: the genetic, the horizontal gene transfer, clonal success, the biofilm formation, and also the toxin antitoxin system. The toxin antitoxin system is actually one of the efficient mechanisms, and it's well studied in E. coli. Yet we thought, okay, like Campylobacter, we never had like something about like the resistant mechanism by toxin antitoxin resistant determinants. So we have some kind of an in silico characterization of novel, like we found actually 10 novel toxin antitoxin systems that are actually uniquely found in Campylobacter, and we characterize this. And actually our next study, as I mentioned, like when the GWAS, you characterize genotype and then you verify with a phenotype. So currently we're actually verifying this novel toxin antitoxin systems in Campylobacter by doing some phenotypic results for verification. Now I just like try to emphasize one of our model organisms that we can use microbial genomics in order to study like the One Health perspective of epidemiological tracing back and transmission dynamics. At a One Health perspective, I hope I could like had some kind of like at least like my slides were understandable. But I mean, like, you know, we have different models as well. I'm usually interested in all zoonotic pathogens, especially the foodborne pathogens. But for this project, so now I'm working also in methicillin-resistant Staph aureus. I'm more interested in livestock-acquired methicillin-resistant Staph aureus and also in different escape pathogens. So for this Campylobacter, I would really like to acknowledge all like my colleagues and key players. It was like maybe a decade of research. I hope I'm like one of the Campylobacter guys, but I would like to thank all those key players, including my supervisors, my colleagues, and also my students. These are the like people who gave us the money over the past 5 years. So I would like also to thank the funding agencies. I would like to thank my team, my undergraduates, my postgraduates, and postdocs for all the tremendous effort they have made over the past years. So, and finally, these are my contact information. Feel free to contact me anytime if you have any question or if you have some kind of collaborative idea. Feel free to contact me. And thank you so much again for giving me the platform to present my work. And thanks, Nabil, for introducing me. And again, my apologies for my 5 minutes delay.
All right, and please let your appreciation be known using one of the buttons on Zoom to everyone in the audience. That was an excellent talk. Thank you very much, Mohamed. I haven't seen any questions in the chat yet. Reminder to everyone listening in, please put your questions into the chat now, and I will relay that First question, obvious one. Can you please share your personal IDs, your contact again, please?
Sure.
Yeah, if you just leave it on this one, that would be great. While that person takes down that take a screenshot of it if anyone's interested, and then you can get in touch. While we wait for a few questions to come in for the chat, I had a few. I was interested in your paper about the local accessory gene sharing, the one with the long title. And I was curious, in that analysis, did you find any genes in the accessory genome that were specific to Egypt, and perhaps you could say something about that niche?
This is a really nice question. Thanks, Nabil, for figuring out this. Actually, this is like, you know, our like one of the drawbacks of this study. Actually, like we have some kind of like potential insight that the accessory genome is playing a crucial role in acquiring more resistant genes. Yet, to inform this, we have to implement GWAS. And like, you know, I mentioned about GWAS or genome-wide association study, but to do GWAS, we have to have a large number of bacteria that are actually sequenced. And as I mentioned, like our Egyptian isolates were not, it was like 112 like genomes that we sequenced. So to answer this in short, we are now doing more sequencing of Campylobacter in order to implement this GWAS. And by doing this GWAS, we can have some kind of association between the Egyptian isolates and the global ones, which are actually much, it's like thousands of genomes. So we can have like, we need just to have a weighted, like, you know, equalities between the Egyptian isolates and the global ones in order to have some kind of insight about what exact genes that are more overrepresented among the Egyptian ones.
Okay. And throughout all of the work you presented, actually, you seem to be alluding that there were specific clonal complexes perhaps that would relate to host or geography.
Yes. Yes.
Was I interpreting that correctly? What are the hallmark clonal complexes you can point to that demonstrate this? Is this something we can generalize to Campylobacter that this is the case?
Okay, so it's what we name it host generalist in Campylobacter per se, based on our analysis, are clonal complex 21, clonal complex 48, clonal complex 45. All these are, we name it host generalist, mean that you can find this clone in chicken, you can find it in pets, you can find it in cattle, yet there are some kind of host species you can find like this, or you can find it everywhere in human as well. So this is that we name it host generalist. And we found in our study that these host generalists are equipped with a better survival mechanisms to be transmitted along this transmission cycle. Yet we didn't determine the molecular mechanism per se. In E. coli, we know that we have a system called the RPOS system. And this RPOS system is like, you know, can actually trigger E. coli to be more cross-protecting or more resistant. But the molecular mechanism why these clones are infecting like this reservoirs is not determined yet.
Okay, I have some more general questions that have popped up into the Q&A, so I will ask those. One question is, what is the biggest challenge when dealing with the One Health approach?
I think like it's as I mentioned, I don't think that like, you know, like it's I can find like there is a challenge myself. It's not challenging. I'm doing the stuff that I love. You know, I'm a veterinarian and I'm usually trying to, and I love microbiology and I love genomics. So, you know, I didn't find anything, for at least speaking of myself. But I think like, you know, like speaking about like, let's make the question more like, you know, like longer. The biggest challenge in working in developing, like in science, working in like in Egypt, like, you know, as I mentioned, like, you know, like you can have a lot of good insights because of like the mixed epidemiology. You know, there is no like, but yet being like the biggest challenge for me in working in this approach is having good funding, because funding is a bit restricted. So like, you know, to challenge like with this type of high throughput sequencing stuff, you have a lot like to secure a lot of money to support, you know. So I think like it's funding wise.
Okay, and I had another question. The participant asks, this may be specific or unrelated to the topic, so feel free not to answer, but how can you see the future of research in Egypt, and what is your advice to one who wants to change his career to join the research space, join the research career?
Okay, so as I mentioned, like to tell you the truth, like, you know, I spent like maybe like a decade abroad, and I thought like maybe spending like this much of time abroad, like, you know, to gain more knowledge about like, you know, so once I have the opportunity to lead, I should be like well equipped with all the knowledge to allow me to lead. So I think like, you know, like I'm always advising my students, just be driven by your passion and knowledge. You know, passion alone is not enough, and knowledge alone is not enough. So if you want to join any of like this research, just be driven by this. As I mentioned, like there is a lot of challenges in Egypt. First of all is I think is a culture of research. You know, it's not about like a well-equipped place. It's about like setting the research culture, which we think like in Zewail City. And by the way, it's named Zewail City because of Dr. Ahmed Zewail, who is a like Nobel Prize laureate and who is the founder of this. So it's kind of like, and why we name it as a city, it's not a university, it's like a interdisciplinary place where you can conduct research, you can have like very good students, and actually have some kind of applied perspectives as well. So I think like I consider myself lucky working at this place because the culture of research is well established. So I mean, like, you know, like for people who wants to join the research, just be driven by passion and also by knowledge. And, you know, like, as I mean, I think like, you know, one of the key success in this is the team I'm working in the institution I'm working in, and also I think like, you know, like the sharing of knowledge among like my colleagues was really helping. So I think like with this type of research environment, if it's like replicated across different institutions, I would believe that Egypt is in the right track.
Okay, I'm not sure if the audience member wants to have a follow-up to that, but feel free to drop that into the chat. I had a technical question, if anyone will will indulge me. With your CGM and ST method that you mentioned, is which panel of genes, or how was that constructed, and is that scheme available for others to use?
You mean the cogenome?
Yes.
Okay, so actually, like there is like different pipelines per se. Okay, there is like a pipeline which goes governs. By the way, I'm not a bioinformatician at all. Like I'm usually like, you know,
okay,
if you're,
that's all right.
Yeah, no, but I mean, like, you know, I know, like I'm just telling my students, because my team is like bioinformaticians, but I'm just telling my team, you have to be better equipped. This is like related to the previous question, you know, with the knowledge, and also, so my passion is microbiology. I consider microbiology my passion, but like, you know, but also like that's what I'm like my team, because they are like purely bioinformatician. I'm just telling them, you know, you have to know about the biology of the bug, you know, in order to analyze the results. Because if you are doing just dry lab analysis without knowing the biology of the bug, it's nonsense. So back to your question, Nabil, like, you know, there are like some pipelines that actually can determine the core genome after removing of the recombination regions. Okay, so this is called gubbins or clonal frame, and the idea of this that it can have a clustering of the core genome, but it actually subtracts like, you know, from this core genome what are the recombination regions. Okay, so actually, like, this is a really good insight. I was mentioned to my students. Another pipelines, it actually add the recombination regions. Okay, so if you do this both analysis, one by gubbins and the other by like a clonal frame, one with removing the recombination regions and the other without, and you compare both phylogenies, okay, and if you found something different in the clustering, it means clearly that the recombination is playing an important role in clustering of the isolates. So this is a basic, you know, biology of the bug. if you compare the both. So I mean, like, this is like what I like, maybe a take home message, that like you should have an insight while you compare. It's not about the analysis. It's not about like, this pipeline is doing what, but it's also like how to use this analysis to study the biology of the bug. So in answering your question about like the core genes, some core genes, some pipelines such as the clonal frame can make a core genome phylogeny, either with or without a recombination region, and another, which is called the Gubbins, actually subtracts the recombination region. There is a lot of other pipelines, like, in the GitHub also, for anyone who is interested. And if you would like, if any of the participants wants to ask about like any of these pipelines or so, please just email me and I will send them like bunch of pipelines for every analysis.
Okay, excellent. And I think we've run out of time. So with that invitation, I will draw the session to a close. And I want to thank Mohamed Elhadidy for talking with us today. And thank you for everyone for attending. Sorry about the late start, but we got there in the end. It was most informative, most enjoyable. So thank you to you, Mohamed. Thank you for everyone for attending. And we'll see you next time on this webinar series.
Okay, thanks Nabil so much, and thanks for Jamie and all like the team for inviting me.